the discussion. You're here, if you don't understand something, to get somebody to explain it to you. None of us are stupid here, but there are some really technical stuff to discuss. And the technical stuff is important because one day the technical stuff will make a huge difference. Because in most severe and persistent mental illness, the core problem is biological. And we need to crack that, as well as the social and psychological and societal manifestations of mental health issues. And you need to understand the technical issues and get people to justify them. And I'll try and do that with the panel as well, not in an antagonistic way, but just in a way of elucidation, so we kind of understand the territory. So, it's going to be neuroscience, standing on one leg for 10 seconds, but we'll just have to, we're not going to be all PhDs in neuroscience by the end of a session. But it is important that we understand. And put up your hand if I let something go to the keeper that you don't fully understand in terms of what people are talking about. On our panel, and these are to thrash out the issues, feel free to enter this discussion. This is not just a discussion for the panel, it's for the whole of room before you break into your groups. Let me remind you that what you're focused on is you're going to come up with one high risk and one lower risk project that could move things forward at a higher rate. You've got a list of things there under that section, the basic neuroscience section in the Delphi, which an international group of experts and locals, as well as consumer carers, have rated. That doesn't mean to say those are the only things we consider. And remember Norman Sartorius's exhortation, if we just came up with one really good one, that'd be a pretty good result for the day. And so you're going to get one high risk, one medium risk out of the day. Partly out of that, largely out of that list, but you may have others that the panel comes up with. But they've got to really make a difference and be doable. Our panel to discuss this are Neil Cole. Neil has been a board member of the Mental Health Research Institute in Melbourne, at the University of Melbourne, for many years, and is a person with lived experience. Paul Fitzgerald is a professor of psychiatry at Monash University with an interest in transcranial magnetic stimulation. Pankaj Shah is our next panelist. Pankaj is at the Queensland Brain Institute and studies some, anxiety disorders and other things, but has develops animal models to study psychiatric and mental health issues. Idmar Khan comes from the Technion Institute, which is in Haifa in Israel, and Idmar, as I understand it, has an interest in neuroimaging and brain imaging, and manages to get amazing images from quite tiny animal models. Cinda Shannon-Weichert is professor at the University of New South Wales and an authority on schizophrenia, and has a wide variety of interests in terms of schizophrenia research. Max Bennett is one of the doyens of neuroscience research in Australia. Is the foundation director of the Brain and Mind Research Institute at the University of Sydney, and his particular interest, research-wise, over many years has been the study of consciousness. And last but not least is Keith Wilson, who we've been introduced to earlier. Keith, just to get the focus on this from a person who's been a carer for 40 years, what are the drivers from your point of view for this discussion?
Thanks, Norman. Well, yes, you're right. I'm not a scientist and I'm not a clinician, and I'm not a researcher. I'm an unlearned person in those respects. But I don't regard myself as totally without a body of knowledge. And my body of knowledge stems from having lived with a son with mental illness, which first showed its signs at the age of 4 and continued through a troubled childhood, school denial, a violent adolescence and early 20s, an eventual diagnosis of schizophrenia, lots of changed medications, eventual prescription of clozapine, which was commended to us as a family, and which, as is often the case, I understand, solved most of his problems about paranoia, though he still has vestiges of paranoia. And as a result of that, and as a result of moving into his 30s, began to go through some sort of maturation, so that now when he's 42, he lives a life at ease with himself, though he needs still to live with me, to have the company and support that makes his life bearable. So what I'm saying really is that I have observed this mental illness for nearly 40 years. I've observed and I've learned from his treatment, his failure to get treatment, his failure to respond to treatment, the problems he has with the treatment he has, which makes him hugely enormous in body size. And in a sense, living with that is uncomfortable, because I know that it would be better if he wasn't hugely enormous, though it doesn't seem to concern him one bit. Therefore, I think that I have a body of knowledge which can stand beside the body of knowledge that you have as a scientist or as a researcher or as a clinician. Unfortunately, my experience with clinicians has been that when I tried to go with my son for consultations, they try to ignore my presence. Some of them even turned their backs on me and pretended I wasn't there. So I've seen that, and I've seen my son walking in the street with his funny gait, and I've seen people turn and look at him. And I know that they don't know what I know about him. So what I know about him is my body of knowledge. And if that's not to be regarded with respect as an authentic source of information for research and for inquiry and for clinical practice, then I don't know what is.
Thanks. So Neil, you've been on the board of the mental health research.
No, I've worked there.
You've worked there? Right, sorry, I thought you were on the board. So as somebody with lived experience and who's been involved in research, and you look down this list and you look at the imperatives, what are the imperatives?
I don't know. It's a big... look, I just feel a bit out of, it's a bit like the story about the three barristers at court. And the first barrister said, I appear for the defence. The second one said, I appear for the prosecution. And the third one said, I appear to be in the wrong court. And I think it's fair enough to say I'm a bit in the wrong court myself today, certainly with this topic. But if we follow on from what Keith said and just trying to put it into context, I work as a consumer consultant, dealing with people with mental illnesses at the now the the Florey Institute. And I think we cross an enormous scope of problems and difficulties. In my own case, having bipolar mood disorder and not being diagnosed till I was 35, would say there was a major problem with not having been diagnosed at 17 or probably 16. And it really is a blight on our system that I was missed so many times over those years. So on one hand, I would say the early intervention thing, which I think is referred to there, is an imperative. I don't think there's any doubt about it myself. You at least should be servicing kids who present with those illnesses. On the other hand, we have a whole wad of concerns with people who acquire depression. So maybe they go through a severe marital breakdown, or they go and fight in Afghanistan and they end up with post-traumatic stress. So there's so many things over broad years, and then of course more and more people getting depressed as they get older. So the issues that I see, I'd prefer to leave it to Paul and others to talk about what the scientific models are, because I really don't understand it fully, but I do think we have to look at it totally and not just focus on the early lifetime, and also that when dealing with adolescents, it sounds absolutely stupid, but I'll say it anyway, is that the most important thing when you're dealing with kids, and I was a lawyer before this and I dealt exclusively with young kids, very few who had mental health problems, most of them antisocial behaviour, stealing cars, you know, the usual things, the occasional murder. And but what I found...
Nothing serious.
No, it was, and he was innocent. But the important point is that the one criteria we should apply to young people is to have some wisdom about how we approach it, so that we should look at their overall well-being, who they are, where they come from, what's their prognosis given their socioeconomic status, their Indigenous status or whatever. So if we do devise models, and we have to, and maybe we have to treat kids who are 15 with clozapine. I don't really like the idea myself, but if we do things like that, do it with wisdom and knowledge, not pushing a barrow of this is what we have to do. But
and we will come back to several of these issues through the other propositions during the day. I think it would be useful for the participants to hear from each of the panellists, you know, in a minute or two what you do in your day job, so that we've got that as a basis, and then we move on to what we could do together, and whether any of these projects that we've got listed are actually worth the candle, or whether there's something else. Paul, tell us how you earn your living.
I spend most of my day either running a series of clinical trials and clinical studies oriented towards really treating the most severe patients with depression and a number of other conditions. Our research program includes patients with obsessive compulsive disorder, schizophrenia, autism and Asperger's disorder, and a number of other conditions as well. And we work very much at the most severe treatment-resistant end of the spectrum. So patients have pretty much not responded to everything else that's out there, be it medication, electroconvulsive therapy, psychotherapy and the like. And the focus of that research is on developing new treatments for those patients, but also doing that in an iterative way where in conducting those clinical trials we gain more information that allows us to improve the way in which we apply those treatments further down the track. And most of those treatments, as you suggested before, are kind of in the brain stimulation space, because the patients that we see aren't getting better with medication and they're not getting better with psychotherapy. So I spent a lot of time listening to the stories of patients who are generally in their 40s and 50s, some of them in their 20s and 30s, but you know, almost universally, there are very few patients that I speak to who don't tell me about something that happened in their childhood, but most commonly their teenage years, as the kind of signal event. I pretty much always start by asking patients, where did they feel it all began? And it's very rarely that somebody will say, well, it started when I was 45 or 33. The vast majority of people, it's in their teenage years. It might be in their early 20s, but there's always sort of antecedents to that. And so when we think of the neuroimaging studies that we're doing, or the other sorts of investigative studies we do where we're trying to investigate brain processes, we're doing that from a perspective of trying to improve the treatments we apply. But I don't actually have any expectation that in doing those studies in somebody who's 45 or 50 or even in their 30s, that it's actually going to give us a fundamental understanding about what caused the disorder to begin with.
How important is it to know what caused the disorder?
Look, I actually think in 2014, for the majority of patients that I treat, it's actually not that important, because I think the complexity of the brain and the complexity of the interactions between people's lived experience, their genetics, their environment, their development, etc., is going to be that the answers are going to be so complex that translating that into something that's going to help my patient tomorrow is not realistic. However, if we actually want to make fundamental differences to the next generation, the generation after that, I think abandoning that approach to try to understand fundamental causes is premature and something I don't think we should be doing. But the greatest fear I have always in these sorts of discussions is that the patients who have been struggling with their illnesses for many, many years kind of get left behind a little bit. We start to only focus on, okay, can we understand, can we prevent conditions? Can we stop it all before it begins? Can we understand what causes this? And we forget that there are literally tens or hundreds of thousands of people with these disorders. And even if we can prevent a third of the onset of depression or a fifth of the onset of schizophrenia or half of the cases of OCD, there are going to be lots of people who ultimately end up with really significant problems, and ultimately having better treatments for those people, I think, is one of the most critical questions, and one which I think we can do things about today without the $200 million of the US Brain Initiative to actually help us understand, how things actually work. I think we're in a far better place to try to develop treatments for the here and now.
So what are your if-onlys? You sit there and you're at the end of your day's work. You've tried certain treatments. Most of them haven't worked. Some of them have helped a little. You're not quite sure why. It's a dark arc transcranial magnetic stimulation. What's your if-onlys? If only I had X, Y, and Z, or knowledge from A, B, and C, I'd be shooting ahead here.
It's certainly in my area, and I think this applies not just for brain stimulation treatments, but I think it applies for a range of treatments that we have. I think the fundamental problem we come up against is the genericism of our sort of diagnoses and our understanding. If I see a patient with depression today and I apply a therapeutic model to that patient, the next person who walks into the room in an hour's time, I think it's fairly likely that their brain processes, not let alone their whole life story, is going to be substantially different. And the things that we do around understanding neuroanatomy, neurophysiology, imaging, etc., so much of that is oriented towards, okay, let's scan 50 people and average their brains and compare them to 50 average brains of people who don't have condition A. And I think that gives us a bit of an idea, okay, this blob on the brain is a bit smaller or a bit bigger, but it tells me nothing about an individual patient. And so taking some of the neuroscience to a level where we can actually understand something at an individual patient level, I think is ultimately what we need to do, to try to develop treatments that are going to be far more personalised as well.
Pankaj, tell us what you're up to and respond to some of the things that Tony was saying.
Right, so... to ask questions?
Yep. Go.
So couldn't a pathway that starts earlier also be a diagnostic tool?
Oh, look, absolutely. I think there's... and I think... ultimately, we're going to have models that include a longitudinal perspective. I have no doubt about that. I think, and that might be an understanding of how did this bit of the brain end up this way it is, but it's going to have to integrate, the bullying they experienced in high school or the workplace problem they had in their 40s that ultimately precipitated their illness. I think there are probably a limited number of final common pathways. The brain goes wrong, interestingly, in a relatively limited number of ways, manifesting in psychiatric illness. We don't tend to walk out, we don't hear patients developing a delusion that the sky is going to turn yellow tomorrow. There's actually a fairly limited and relatively stereotypical way in which the brain goes wrong, but I actually think there's probably a whole vast of ways of getting there to that problem.
Pankaj?
So, I'm a cellular biologist and run a lab at the Queensland Brain Institute, and we're working out circuits. So Max talked about connectomics last night, and I agree with Paul in the sense that, look, many of the disorders that we're talking about here are very complex, and trying to work out the circuits is perhaps a bit premature. The view that we've taken is, well, it's likely that the brain works in similar ways in lots of different areas. So let's try and understand one bit of it which does something that we know about. And the view that we do understand how the brain works is just not true. We're just scratching the surface of what the brain does in terms of the computational skills that it has. So the circuit we work on is the circuit that processes fear. And this is, we use very simple paradigms. And
And just for when you say paradigms, you're talking about animal models.
Animal models. And the beauty of this system is that the same animal models work in people as well. You can do the same you can put the same stimulation in
so
people.
You can scare the shit out of me and I'll respond the same way.
In the same way, exactly. At least from the physiological measures, right? And the circuits that are involved in this are probably pretty similar as well. So what we're doing is we're going to try and figure out the circuit which mediates this response and how, when things go wrong, do you treat them. And it's pretty well accepted now that, you know, in things like anxiety and post-traumatic stress, somewhere in the circuit there's something wrong, because it's what's wrong is processing our thinking about fear. So one of the propositions here is, you know, what a neuroscience explanation. So what constitutes a neuroscience explanation? So is that you do an fMRI on a bunch of people and say, oh, in these people, you know, we see these effects. And what's come out a lot of this kind of stuff is risk factors. And so what we're looking for is not a risk factor. We're looking for something like what Julia talked about, you know, Helicobacter. Here's a bacterium that infects. You fix the bacterium, you fix the problem. And a lot of the therapies that are out there for most things are treating the symptoms. They don't go to the heart of the problem because we don't know what the heart of the problem is. So we use animal models, and on the other side of the coin, you know, we go in with people, same sort of people that Paul's working with OCD, with depression, and these people are having electrodes implanted for DBS, deep brain stimulation, and we go and record from these people's brains, and we do very similar experiments in theatre. So we get them to do stuff and record the activity, again trying to work out what kind of circuit dysfunction is there. So my view really is a very reductionist view, and I think what we should be doing is picking some part of the brain which is involved in one well-characterized disorder and try and figure out, you know, what kind of things go wrong there, what exactly is going on in the brain. And you do this by doing the cell biology, figuring out the circuits, the receptors that are present in detail, which is what we do, you know, work out the subunit stoichiometry. Then you can design drugs which target those specific regions. And so, you know, I think we need a better understanding of the neural mechanisms at some level before we can progress.
And just to you know, translate the jargon, stoichiometry is how the signal binds with the receptor and makes the signal happen.
Exactly.
Now,
look, this is, you know, I agree completely. Look, the way things are right now, we need some way to treat these people. There's no doubt there's people out there which would need help. Yeah.
You know, the animal model is one of the core issues here. And so one of the problems that's held up cancer research is that you go to labs, you could go to all the cancer labs in Australia, and each one's got a different animal model. And they all love their animal model because they think theirs is the most important. And so they go along a line and they've got a brilliant idea for a new molecule that might affect this particular cancer. Doesn't work in that animal model. They come to the end of their grant. People go off to the four corners of the earth. And then somebody starts on another animal model. And 20 years have gone by because of people's favourite animal models, which might have no relevance whatsoever. We've killed thousands of mice and without anything happening. How do we speed? So if your presumption is right, that we go from animals to humans and back again, how do we do this faster?
Well, I think,
to get more meaningful results and stop all this bloody fragmentation.
Well, Noel, if you think about the DNA world, right, way back in the 40s when they had that meeting, a bunch of these guys got together, or women as well, and they decided they're all going to work on one bacteria.
One woman made the difference, actually.
Yeah. No, this was the structure, right? But in terms of working out the genetic mechanisms, they all agreed to work on E. coli. Right. And I think we need some sort of agreement like that to say, look, we're all going to work on this one system, basically. And there's no shortage of space and things to do. It's not as though it would be, you know, sure, it would be competing, but that's a good thing.
So you roll the dice, you bet the farm on one system. That's a hell of a thing to do, isn't it? In a complex thing such as the brain.
Well, not really. I mean, you know, it's a pretty complicated place. You have to start somewhere. And I think focusing on schizophrenia is you know, despite that you do need treatment for it, right?
If I were to say we're going to study Perth by studying Joondalup, you might get yourself into some problems here.
No, but it's a question of what it is that you want to study, right? Supposing you're interested in what kind of plants are present in Western Australia, that's not a bad way to start, right?
Okay, well, we'll come back to this. Inmar, you're into imaging. Just tell us what you do at the Technion.
Okay, so I'm a system neuroscientist. What we're interested in the lab is actually using both humans and animal models, mice specifically, using fMRI. It was mentioned here before, functional MRI. So we are looking at functional MRI, to those of you who are not familiar with the technique, is basically measuring changes in metabolism in the brain. And the assumption behind it is that if we have a bunch of cells in the brain that are computing something, they'll use more energy, more oxygen, and we are going to pick up this change in metabolism. And some of the work that I've done before actually starting my own lab was to validate that if you measure a mouse's brain with fMRI, the signals that you'll get, both spontaneous and signals that are evoked externally, are similar to those that we see in humans. So we kind of established it as a translational bridge. The machine
so you can
itself,
find out which football team the mouse barracks for, can you?
Something like that, or fear, or if it actually sees some visual input or sensory input. If it's brain, if the functional MRI signal that we are measuring is actually identical to the one that we measure in humans. And the reason it's that important is that mouse's brain, probably most of you are aware of it, are different from human brain, and they are different on so many levels that one thing that you can question is if you actually measure with the same technology, you might be measuring something completely different. So we took the effort of actually kind of validating that little bit of information. And the reason why I think it's important is that now we are positioned to look at, we are kind of very, very interested in intrinsic signals in the brain, so signals that exist in the brain in the absence of any external input or output. So something
so when the brain is just ticking over.
Yep, so it's doing its maybe maintenance, maybe actually, what you would consider thought. Okay, so both in mice and in humans, we have these spontaneous signals, and we're interested in looking at these signals across development, for example, so something that is relevant to some of the discussion here, and understanding how development occurs, what happens if you have some disruption during development, and again, looking both in humans and animals, so in mice. So trying to kind of, take a comparative approach, so compare between the species and try to understand the similarities and differences.
What have you learned?
So what we learned is that the sensory systems in the animal and the human, they look very much the same. Some of the
when we talk sensory, we're talking about hearing,
visual,
feeling,
audition, somatosensation. We think that there are fundamental differences in high-order cognition, as expected.
Cognition being thinking.
Being, for example, what would be the precursor of language in a mouse? Okay, do mice have some kind of language? So they have these ultrasonic vocalizations. So obviously they use audition, auditory stimuli to communicate something. So is it similar or different from humans? We think that it's radically different. So this is one example. We think that there are
we could have told you that first.
I'm sorry?
We could have told you that beforehand, I think.
But still, it's good to know that it's actually the case, right?
Yep.
And we know that other systems that might be very, be seemingly very complicated, like the fear processing, are very similar. Okay, so now you can have something that, the instantiation in a human would be a very complicated one, right? So, what would be an anxiety in a human and anxiety in a mouse? They can be completely different, but they are driven by very similar subsystems.
So I've got a couple of questions to ask, just sort of validating kind of questions. In clinical medicine, if you show, a bank of cardiologists an electrocardiogram, there's significant differences between them, showing them exactly the same ECG trace. Show radiologists the same X-ray, they will come up with significant different interpretations of that X-ray. So you're looking at very complicated images on fMRI. Mm-hmm. So before we put all our money into fMRI, how reliable are, and it comes to Max's point and others about seeing what's before you, how reliable are your observations? So you've got this very expensive technology, but if you look at it and somebody in Stanford looks at it, are they seeing the same thing?
So I think that the functional MRI is a relatively new technique. Okay, so it exists for about 20 years. which counts as a new technique. Its first years were hard, and I think that they produced a lot of spurious results, or results that one lab saw and never repeated again. Since then, I think that we've accumulated huge amounts of data that now you can corroborate a lot of the the basic findings across different groups. And I think
so. If you find something in Haifa, it's replicable in Stanford.
Completely, it's replicable. So now there are, for example, the human database, the kind of human imaging databases that we are using. We're not collecting the data ourselves. It's being deposited in databases across the world, and you can just download it, and you can run whatever analysis you want on it. There are thousands of subjects by now, and we are starting to do the same thing with mice. So actually collecting the data and kind of publishing it, and this is also hundreds of samples. So it's not something.
So you're going on and you're making fundamental findings about how the brain works and extrapolating that to humans and back again. And you may or may not find something that helps in unlocking the mystery of some chronic and severe mental illnesses. But in 2014, if you imagined a bigger project which uses your knowledge and technology, and also Pankard's, and also the clinical knowledge and clinical research expertise of someone like Paul, and we haven't even spoken to Cindy and Max yet, what could you contribute to a bigger project without necessarily distracting you too much from your own research, from what you know, that you could say, look, here's a project where I've got this in my saddlebag, we can make a difference?
Yeah. So I'm going to read from your, from the proposition itself, because, I would assume that some people either haven't read it or forgot, and I think it's important, and I'll answer using that question. So the citation is, the most clear signal from the vast body of work in humans is that the major psychotic and mood disorders present in their distinct forms in the post-pubertal and early adult development phase. So that's from the proposition, and Julio spoke about the pink elephant in the room. I think the pink elephant in the room is we don't understand the developmental processes that humans undergo during those stages. So really, if we, from my perspective, what we need to do is we need to think of ways, and I have one suggestion, but obviously I think that's Okay, so the suggestion is actually to use imaging, human imaging. So this is something that will not distract me from my work because I do mouse imaging, but it will benefit my work because we are looking at humans. Do a lot of human imaging, especially individuals that we think are at risk for psychiatric disorders, people that we think that clinicians observe them as being at the pre-stages of an outburst, so someone that might be kind of prone to that. And I also believe in just collecting a lot of data in that perspective. If you collect a lot of data, if you do it in a standardized way, I think that we're going to have a body of knowledge that is now accessible to scientists, and in there we could identify processes that are relevant for to understand development. And I think we need to understand development.
So I understand. So just again, I don't want to get too much detail too early, but do you have to follow a single person through life, or is it sufficiently reliable from your knowledge now that slices through age groups would give you the knowledge you need? So I think. So in other words, do we need to wait 20 years, or can we do it in a year?
So I think that if the numbers are large enough, we can do it, cross-section. We don't need to track the same person for a long time, but I don't think it's guaranteed. I think that there is a process here that might take more than 1 year, and we need to get ready for it. Also, another aspect of collecting all this data is someone needs to look at the data, and that takes time as well. So I don't think really there is a shortcut here, but I think this is actually, when looking at the projects, this is the number 1 and the number 3 rated kind of in being valuable and feasible.
Let me just explore the skill base. I was part of a discussion in Victoria, the developing a new comprehensive cancer centre. And the director there was determined to bring people in, exactly as Helen said, who'd never worked in cancer, outsiders, to try and shake the tree. And a cancer researcher, an eminent cancer researcher at a leading cancer research institute, was moaning about a computational problem that she had. It was actually he had, and nobody could solve it. And somebody from computer science says, I can do it for you in 10 minutes. We solve that problem every day of the week. And in 10 minutes, as a result of a chance, this is serendipity at work, of a chance meeting, the problem was solved. Who do we need at the table who's not at the table now, so that you've actually got the mathematics, the computational techniques, so that in fact you've got this data and it is being utilized and you're moving forward, rather than scrabbling around 5 years later saying, oh, I wish I had, you know, a mathematics group or a bioinformatics group.
So I'm not sure we have the data, to be honest. I think that we have not acquired the data to tell us about developmental processes in humans, I mean. So I think that there's still missing link there, and I think that acquiring the data, even without, you know, this is a part of the idea to put it in a in public database. It's actually you can then harness people that don't have access to the type of data, don't have access to actually the resources to acquire the data, right? So, and I think that then you could use people that are, I think that there are mathematicians, there are engineers.
But wouldn't they ask different questions at the beginning of the process rather than halfway through?
So that's true, but I think that some of the, you know, the method that I'm referring to is the intrinsic signals in the brain. People call it resting state. It's actually intrinsic signals that exist in the brain. I think that they contain, it's like a DNA in a sense. I don't think, you know, it's possible that it won't be the whole story, but I think it contains enough information that we can make headway just with those signals. And really, realistically, what it means is that you take a person, you put them in an MRI scanner for 20 minutes. This is all you need. You get all the anatomy and the functional imaging that you need in 20 minutes. So this is not, you know, it's a conventional scanner. So really, just that data in enough people, I think that, you know, it has to be strategically decided which and when and all these parameters. But then making this data available, I think that will, in a couple of years, will give enough kind of infrastructure for folks to make discoveries. But we are not there yet. I don't think we have the data.
No, can I just look at the QBI? So we've been doing this other stuff I've been talking about, but also combining fMRI and the teams that we've assembled. It's become pretty apparent in the last 2 years. You always need computational people on board. For a start, you don't know what you're looking for, and the signals are very data intensive, and you need techniques to analyze this data, which most of us just don't have those. So I'm also part of ARC Center of Excellence now for Integrated Brain Function, which is bringing together people doing fMRI, sort of stuff we're doing, but also has a strong computational focus in that as well. See, I think for this group, we definitely need some of those people on board.
But how do you integrate real life into this? I mean, I understand. I mean, I know I'm being naive to the nth degree, but real life. I'll give you an example. I recently brought together a group of pancreatic cancer researchers with outsiders in the room. And two of the outsiders in the room were computational people from big data, from retail, a company that's half owned by Woolworths. So they know more about us than we know about ourselves. They know what magazine you buy if you buy a mango. And so they're dealing with huge amounts of data. So they're sitting there, and a world authority on apoptosis, programmed cell death. So those of you not technically in the audience, one of the reasons you get cancer is cells don't die. So this guy, he's Walter and Eliza Hall Institute in Melbourne, programmed cell death, and he's talking about genetically identical mice and how they behave differently, and how it's a random effect. And so the guy who started this $2 billion company, who's a statistician, said, excuse me, I didn't understand a word you said until you used the word random. And he said, bullshit. He said, if you're using the word random, you're covering up for your ignorance. Nobody in my company is allowed to use the word random. There's no such thing as random effect. So this guy went white, absolutely white. And it was one of those moments that Helen was talking about, which is he wasn't angry, he realized what he'd been doing. And I mean, there isn't an argument for randomness, but so just having somebody else there, and what he can bring into the room is when you've got your brain scan there, he can tell you whether that person ate too many Rice Krispies in the last 3 weeks, which may have an effect. I'm being bizarre here, but may have an effect. So the question is, have we got the right people round the table at the start so that we're potentially able to do that? Max?
Can I just comment on that? I have two superb mathematical teams, not represented here, working on these problems, and they all come from bioinformatics. That is, the mathematical analysis of the nucleotides or the genes, if you like, that make up your genome. this is a well-established field. And it turns out that the mathematical techniques developed in that field, a very mature field, can be utilised in this area of trying to understand these brain signals. So we have an economy of experience, and we have people who are very gifted in these areas and don't have to know who has got schizophrenia and who hasn't. That's perhaps my job or my clinical colleague's job. But if you can bring these teams into the game, I think you can make significant progress.
Yes?
I was just wondering, you were saying earlier that you can sort of get a picture from just getting a person from different age groups, maybe. As was said much earlier in the piece, how would their lives... how would their lifestyles, experiences and all that would change where they're at today? So, you know, individually. So how would you work that out to get to where you want to be?
So I actually think that you're pointing out something that might be a huge kind of disadvantage of that method, and would be completely averaged out, right? So we'd actually miss those individual differences with that approach. So I don't think it covers all the...
Except that it's an excellent question, which I think Idmar's acknowledged. But in cancer biology, they've solved this problem. That if you take a sample, it's got to be fully clinically annotated. So it's got the person's history attached to it. So you know what the MRI means.
So that's been done. It's like the phenotyping, the kind of putting all the information, handedness, and a lot of the information is actually being included, and you can factor it in. I actually should, Norman, I should say that in the teams now, I think that most imaging labs, they have mathematicians, they have computer scientists. It's true for my lab. If you go to my website, you'll see that I have a mathematician, I have a electrical engineer, I have a physicist, I have physicians. So I think that they're already sitting in the table. Maybe I'm a mathematician by training, so my undergraduate degree. So I think we might...
We're impressed with your social skills already.
It took me 2 decades.
One thing I think is happening is we are not doing enough cognitive blending. And we are not talking about the system you are using in your lab is not talking to the system is used in Sydney. At the moment, I don't see why you cannot continue collecting your data and somebody's collecting biomarkers. And somebody can go to the same system, a clinician, put a history of the person. It could be.
So what would that integrated system look like?
It has to be a little bit like how we work with done different labs around the world. I mean, some people can get together from different...
So how do you deal with the problem here? So as soon as you say that...
language.
No.
Only speak English.
But it's been done, Norman. I must say that it's been done.
Right.
The databases are available. I'm actually, using a lot of data that is not mine.
Yeah, but you're not using data with biomarkers, biological markers.
No,
like looking at the brains. If I wanted to look at your data and I wanted to put things, I want biomarkers plus your data plus history.
What kind of biomarkers?
Data at once.
What kind of biomarkers?
So just to explain,
inflammatory.
Just need to explain. So, but when we're talking biomarkers, you'll hear this word used a lot for those who are consumers and carers or not from this area. We're talking about things that you would measure in the blood, or even in images actually as well, which indicate that you may have a certain problem going on, or you may have a certain genetic profile, or things like that. Or, so it could be your white blood cell count, it could be your cholesterol level, or it could be a genetic profile.
I suppose what I'm talking about is that I think what you're doing is wonderful, and I think it's extremely important. But at the moment, what we are not having is integration of knowledge. We all are working...
So I think Idmar needs a right of reply, because I think he thinks he is doing some of that.
So actually, I completely agree with you. You say biomarkers, the brain is also kind of a biological system. So when we image it, I think of it as a biomarker, maybe a different dimensionality, so more data. But I actually think that DNA, people are adding that, other information from the blood. These are all important. There are, the United States is with every everything in science, is leading the way in databases that contain that information, and also building infrastructure that allow you to analyze the data. And I think that, I hope that in Israel or here we could, do something like that. And one of the things that I wrote for myself as a note for this panel is big data. I think that we need to have big data. We need to have the resources to accumulate it and analyze it. And I think that this is something that I'm fully
Julio, and then Michal. So I'll just come to Julio first, then I'll come to Michal.
I have a comment here, which is comment and question at the same time, but it is a question. In my more clinical years, I'm just thinking of a specific case here. I remember vividly this older teenage boy, like from university, who was from a wealthy family, was hospitalized, was getting psychotic, and it was very clear, initial onset of schizophrenia. So he was not on drugs, he was not didn't have psychosocial stresses, was at university, and then he's hospitalized and starts to hallucinate, and you can see the kid walking in the hallway, talking to the voices, speaking back, and clearly, psychotic and, kind of a floridly schizophrenic. And it's something so visible, it's nothing very few things in medicine are so dramatic. And you take a blood test, there is no blood marker we can find. You do all the imaging in the world, there is no image marker. You can do whole genome sequencing, genotype everything, there is no gene marker. What is it that that is there in the disease that we just cannot see, but yet it's so drastic and so visible and so dramatic?
So yeah, maybe I should let others respond as well. But I'll just say that actually, it's what you said before. The answer is probably there, but we don't see it, right? So we are missing it, and I propose to look more.
So Cindy, your response?
Well, I think that the way that we've been trying to approach biomarker discovery has been problematic because we haven't thought of the disease as separate entities. We know there's multiple causes into schizophrenia, yet we look for a gene that works under the umbrella of the diagnosis of schizophrenia versus controls. I think when we start to appreciate the heterogeneity and incorporate that into the design of our studies.
So heterogeneity is a term which you hear frequently, which means differences, there's multiple differences in the situation that you're looking at rather than homogeneity, which is the same. Sorry, carry on.
Yeah, so I think that's one of the things that's held the field back. Even though we've known about heterogeneity for a long time, it doesn't seem that it's always translate into the research design.
So just tell us what you're doing and
Okay, well, first I'd like to make a comment about the conference so far. What I really love about it is that it's actually valuing the perspective of the consumers. And I actually have lived experience with schizophrenia as my twin brother was diagnosed when he was 17. And I was motivated to try to do something about this disease because I saw him suffer going on and off various antipsychotic therapies. And I didn't really share this with anybody, in part to protect my brother. At the time, it was in the early 80s. But also, as I became a scientist, I didn't want to share it because I thought that the other scientists would view me as somewhat not objective. And one thing as scientists, we're supposed to be cool and analytical and not motivated by our emotions. So that was a very difficult thing for me to sort of keep silent about. And it really was when I came to Australia, got the distance, perhaps the maturity, that I started to share it with the world. And it's been a hard thing, but I'm glad that I did that. But when I actually found out about my brother's illness, I didn't view it as that it was my brother's fault that he had schizophrenia or my mother's fault, but that his illness had a biological basis. And I thought, well, I'm going to go in there and figure out what's wrong with his brain and then fix it and get on with my life. How naive was that? That was 30 years ago. So I decided, because I viewed schizophrenia as a developmental disorder, so I was witnessing the disease unfold, and I was guided by those clues during my education and training. And so I viewed schizophrenia as a developmental disorder, one that emerged during adolescence. So I decided to become a molecular developmental neurobiologist, and I made many discoveries at the bench.
And I want you to try and explain what that means.
So, you talked about the importance of understanding adolescent development. That's exactly what I did by studying the postmortem human brain of children throughout the lifespan. I've made, dozens of papers on this. I don't think anybody's reading them, but there's like a whole transcriptomic database on how genes change over the course of development and what's different about the adolescent period, but it doesn't seem to be incorporated into the latest thinking about the uniqueness and the vulnerabilities of the adolescent brain. But perhaps one of, I think, my major contributions to the field is that I found that there was a mutated or altered hormone receptor in the brains of people suffering from mental illness. And I, again, found this in brains that were donated after death. So once I figured out how this receptor was broken, I could then try to test ways to fix it. So I transfected it in cell lines, was able to find a therapeutic that was used for a different purpose that overcame the broken receptor. And then I collaborated with my husband, Tom Weichert, to reposition an available drug that was already developed for osteoporosis, so it was known to be safe in humans. It worked on this molecular pathway, and we went straight into clinical trials, which we just finished within the last few months, and I'm happy to report that we saw improvement in our primary outcome, which was memory and attention. So I think that that's an amazing place to be right now, and I think having those insights from my brother's illness and his suffering has changed my perspective and the questions that I ask at the basic level, and it has led to a certain level of success so far, at least in our preliminary clinical trial.
So what are your... So apart from other people reading your papers, what are your if-onlys?
What's that?
What are your if-onlys?
I think that people have to recognize the role of things outside the brain. I think one of the problems with neuroscientists is they're so brain-centric. And, there's this big hype to map all the connections down to the synapse. And I think that, we have to remember that the brain exists in the body. There's the hormones that are involved, not only sex hormones, but stress hormones, and that the immune system is in constant interplay with the brain, and that the person exists in that larger setting of I think we heard it referred to as psychology, but it's amazing that you think the mind can be separate, because when you influence things or when you experience things in life, like psychosocial stress, ultimately that changes physically the brain. So we have so many clues right now about how we can go forward, like understanding early life stress in our models and in maternal immune activation. And so I think what if is if people sort of put the brain in a context, I think we'd be a lot further along.
But so and let me just talk. So you've discovered gene patterns and gene expression patterns, how genes translate their message into proteins and messages in adolescence.
Well, that wasn't my discovery. I think that's the central dogma in molecular biology. But yeah, I've actually done a lot of work on looking at how molecules change across the lifespan.
So you're finding patterns of switching on and off in adolescence.
Yes.
Do you know whether that tracks through to adulthood or whether that changes? And are there clues to treatment in there that have been not exploited? Coming back to the fundamental proposition here?
Yes, definitely.
So what would it take to have a project? What would you need, or what sort of group would you gather to start capitalizing on that information?
Right, so I was really taken by the concept that Julio talked about with this war on mental illness, and I think what we really need is a trained army, which we actually don't have here in Australia. So I think one of the really important things would be to take the psychiatrists and teach them more neurobiology, take the neurobiologists and teach them more psychiatry, make this part of our educational system where we have MD, PhDs working shoulder to shoulder, where we have medical doctors with more research training. I think that it's not something that happens
But every man and his dog, sorry I shouldn't use that word when I've got Helen Christensen in the room, but they're all developing brain centres. You've got one in Melbourne, you've got one in Brisbane,
But we're not
soon you'll have
changing
2 in Sydney.
We're not changing the culture. It's just what, like Julio said, you know, you've got these panels of basic and these panels of clinical, and there's not enough bridge people. And oftentimes, I think in the Melbourne Brain Center, they've got the basic scientists in one building, and then they got the epilepsy program in a completely different building, where you have Ian Hickey's idea and Max Bennett's of the brain and mind, where everybody comes through the same front door and they work together because we're working together on the same problem. I mean, the consumers, the carers, the researchers, the doctors, that we need more of those things that are integrative.
Michal.
Sydney, you raised the issue that I was about to raise. We tend to forget that the brain is part of the body. And I think that I absolutely agree with you. And one of the teams that you do need is to bring in immunologists into the team of those working on the brain. The brain without the immune system doesn't work. And you can try to induce an animal model for schizophrenia, developmental schizophrenia. You will get totally different outcome if you induce it in immune-compromised animal and immune intact animal. So it's absolutely part of the developmental profile. And back to your suggestion, Itamar, if you follow up the patient and you say, I have all the information, without information about the immune system development, which change around puberty, it's not all the information.
Yeah, absolutely. I think that's critical, and I would also add endocrinologists to the team as well. So in terms of changing things, though, and what would be the big project I think that we should take on board, I think we should really think about Australia's position in the rest of the world. And we should look to Pat McGorry's leadership in early psychosis and early intervention. I think we should build on that, because the rest of the world is actually looking to us to develop these algorithms, not only to predict who's going to transition, but to predict who's going to respond best to which early treatment. And these can be benign treatments like fish oil, or my favorite, which is this hormonal intervention based on estrogen receptor therapy, or anti-inflammatories, and run a parallel clinical trial nationwide where we integrate the clinical history and, you know, cognitive testing, brain imaging, and blood biomarkers, and it all goes to a central repository that we data mine, and we actually develop treatment algorithms. So we have something to teach the doctors about what to do when we see a patient early on in the course of the disease beyond antipsychotics.
So a massive sort of national project. Fiona,
Yes.
and then you've been waiting patiently. I'll just come.
I wonder if we haven't got an incredible opportunity here to solve a few of the problems in terms of data and how it might be used. And that is that we have now a large number of ongoing cohort studies, all of whom are maturing into adolescence and beyond. We have one here in Western Australia, only 3,000, but they're now 24. We started in utero. We're part of an international collaboration for things like asthma research, things like, you know, gene interactions in some of the other things. Well, we might just be able to, as an international initiative, use the information by doing functional MRIs, by doing biomarkers. These kids aren't too young yet, aren't too old yet for us to start doing that. It would take a lot of money. But we've got a lot of information on these, including the lifestyles, including how they feel about the world. These kids are actually very excited about being parts of these cohorts and tell us a lot about what they are worried about. A large number of them actually have mental health problems already. We're talking about the Bristol cohort, the Brazil cohort, the cohorts in Sweden and Norway, the cohorts in Canada, as well as Australia. So put them all together, there's a significant numbers. And I think that, you know, it'll take a lot of analytical grunt, but we could look at a whole lot of additional data collection immediately to address some of these issues. And then we might even be able to do randomised control trials with these kids who've got these problems. Might not be enough numbers, but to actually start looking at that, I mean, I don't think there's going to be enough. So I think that's perhaps
I
not
think that's
so
fantastic,
useful.
but I think I would start perhaps nationally, and because if you look at other, you know, countries, like I was just at the Society for Biological Psychiatry, and they started on their prodromal research, which is actually, you know, prior to getting first break of schizophrenia, probably 20 years after Pat McGorry, but they actually have cohorts that are as large, and they're developing these, you know, prediction algorithms, so they're going to just steamroll ahead of us, which is, you know, too bad, because they actually have federal support for these nationally integrated studies. And I think in Australia, because we actually are ahead of them in some ways because we have a precedence for record linkage. They don't have any record linkage going in the United States. They don't have coordinated ethics. So
just hold the front page for a moment.
Yeah, sorry.
You're talking the language of scientists, which is competition, rather than rejoicing in the fact that they're ahead of us and what can we learn from them and actually use that so we don't actually have to repeat what they've done.
Yeah, no, I think we definitely need to collaborate, and I definitely think we need to learn from what they've done. And in fact, Pat McGorry is in conversations with the head of the NIMH, you know, as we speak probably. So that's definitely on the equation. But what I guess I'm saying is we don't want to lag behind because the rest of the world was looking to us for innovation and for a leadership position. And I think it's great that they're on board, but I just don't think we want to not have a voice or a position at the table, because I think that we could do better if we integrated, because I think there's too many silos within Australia itself.
Number one, Cindy for General.
General what?
General in the war.
Right.
Chief of Staff. And when you do slice cohorts, how do you allow for cohort culture difference, like baby boomers versus Y generation and so on? What are the practicalities of that and comparing cohort slices?
I think the expert in cohorts is right behind you, so we'll just get Fiona to answer that.
It's a really good question. It's a really good question. That's why I like total population linked data, because you've got an ongoing cohort. Every single birth cohort is enrolled in your linked data. What you've got to do is link all of your cohorts into your population data so you can see how biased or whatever. But it's a really good question. And what you do is try and get as many staggered cohorts as possible. Now we have, you know, UK biobanks. We've got big data sets around the world. Wouldn't it be fantastic if we could bring them all together? And then that would get over the problem that this wonderful lady's just commented of using different eras.
Yes, I'll take your question and then up at the back there.
Yes, thank you to all the panellists. You're a talented mob and we need you. But coming from a family perspective, it's not General Weikart, it would be Genghis Khan that I would like to lead us. But you know, when you're collecting your data, are the families included? Because it certainly hasn't happened in my case. You know, we have a carers charter here, a families charter. So you're asking families for their permission or to be included in the data collection? To be included, the families' perspective to be included. Because from years and years ago, when I knew there was something not right with my child, and we don't know the scientific reason, parents don't know, but you know this kid is very different to the other 3 kids. There's something not right, anxious, not happy. Professor Fiona Stanley has said many, many times, we should be working with families, young families right at the very beginning. Trips to the GP, there's something not right here, there's something, no, it's all right, it's just, yeah. We know a lot, and you need to include us because thousands and thousands and thousands of dollars have been spent on my son, and at the moment I've been told, oh, he's in the downward drift. Downward drift? No hope there. And, you know, data collection must include what families see and live with every day. We can make a big difference.
So, Raphael?
Can I comment on that? I mean,
yeah.
I think that we do try to take into consideration what the families say. I mean, at least from my perspective, I speak with carers and consumers groups. I interview the family and the person. I ask them, what's their biggest problem, the consumers? And they often say, it's my sleep or weight gain or, you know, and those research agendas need to be taken back to the scientists and so that we can try to do something about it. I think oftentimes there's a disconnect.
So the question, I think, is integrating that in a systematic way. So, you know, to use the analogy that Idmar, you know, you have a bank of fMRIs, or you do complicated receptor studies, such as Pankaj does. So the question then is, so incredibly complicated data, but you're already doing complicated computation. Why don't you annotate those stories as well, so you can get those linkages too? And not a lot of that is actually being done, unless I'm wrong.
Actually, you know, first I want to acknowledge that about the biomarkers and the immune system, I think these are crucial. You know, I'm a neuroscientist and I image the brain. I think we need that data. We need to take that data, so that's important. But also to the incorporation of the families, I think that the barrier is not a scientist. A scientist actually would love to, you know, to get that input, to get it as early as possible. In Israel, I know that it's the, you know, the healthcare system, which actually, you know, it's expensive. It doesn't want to support that. And this is where I think philanthropy can help a lot by actually sponsoring some of the, you know, the cost of these imaging studies or other taking of biomarkers. So I think the limitation there is not, you know, scientists, we want that data. I actively write in my grants to acquire that data, and it's being blocked when you talk about humans in Israel, it's being blocked by the healthcare system, which has its own kind of agenda, and that's completely separate. So I think that, you know, the scientists are not the limitation here.
Props to you for putting it in the application.
But it doesn't help either, Norman, to talk about them as stories. They're not stories. They are data. They are information. They're not stories.
Yes.
Yes. Look, I'm
sorry, and after you, I want to come to Raphael here.
I'm a rabble-rouser and not one of the usual suspects. As Keith knows, I make noise. And one of the things that I'm very interested in is that intergenerational mental illness does not come into the picture very often. Now, we have 4 generations of mental illness. I have an identical twin sister, and if you hear her voice, it's not mine. But it's very much like mine. She's in the room here. I have been totally neglected by the mental health system. My mother was. We looked after my mother from a very early age. She had mental illness from the age of 16. We had massive child sexual abuse, ritual abuse, and all sorts of things. Now, what astounds me is that they call it mental illness when it's really trauma. And yes, I do need to take medication, I know that. But it's taken me 69 years to unravel my life, which I'm only just beginning to unravel through a psychiatrist who, 5 years ago, took me seriously. And because of it, with domestic violence and all the rest of it, and fraud from my ex-husband, which I've now got the federal tax people onto me, by the way. That's okay. I don't know how I'm going to get out of that one. And loss of house and loss of my children. 6 grandchildren I don't see. And Neil, I'm reading your book. And yes, I think I had bipolar. Lovely, it's fantastic. I'm having a real crack up at night in bed. I think I would have had something like bipolar when I was quite young, and I certainly had anorexia at school because I used to feel sick every time it came to school dinners in England, and anybody who's been in England with school dinners would know what I mean. But I have a question and a suggestion to the board or the panel. When are we going to look at real human beings and not numbers and data? Because we have children out there, and one of the things that really concerns me is about parents who have mental illness. And it concerns me when I see our Commissioner for Children in the paper with a flippant remark saying parents who make wrong will put IUDs in them to be able to get their parent money.
So can I just pick up on what's an important question? I just want to ask, because it is about the tools moving forward and whether we have them. I'll actually ask Paul and Keith's challenge about data. So talking about real human beings, real people with real experiences. And if I could get the microphone going to Raphael, please.
Excuse me, Norman. Is it right if I just butt in about data, or is...
No, can I just ask the question first, Travis? do we need to develop the tools that turn the experience into data which allow measurement and correlation with what's being discovered in biological laboratories?
I think it's a challenge, and I don't think there are many ways in which it's been adequately addressed today. You know, there's a good example in San Diego at the Brain Observatory where they're collecting post-mortem data on individuals who, during life, agree to donate their brains when they die, who might be afflicted with various conditions. They get brain scan during life, but also they get extensive historical data from those individuals. You know, what are their likes, their dislikes, their life experiences, etc., etc. And that's all being stored in a very data dense manner with all the biology and all the sort of the history of those individuals. But I don't think we really have got yet conceptual frameworks about how we're actually going to tie all that together. But it matters if you have, they're coming at it from a neurological disease perspective. If you have one person who's died and donated their brain to that database, who's had motor neurone disease that starts in their legs, how is that person different from the person who dies from motor neurone disease that starts in their arms? And so instead of considering their two brain scans as equal, they're both brain scans of people with motor neurone disease, they're conceptualising that, well, what is different about those two brain scans? About, how do these people differ? I think we need to be asking similarly complex questions about psychiatric disorders. And I think that one of the critical questions about the imaging, the longitudinal imaging, is that if we go ahead and follow people for their, during their adolescence and their development, we're not going to get the answers at that time. We're still going to have an even longer perspective because, if we scan thousands of troubled adolescents, we actually won't know what the outcomes of their mental health problems are until they are in their 20s, their 30s, their 40s. So we're going to have to accept that, to really get a fundamental advance on this, it's going to take some time.
Rafael, and then Julio again, and then I'll come to you.
Thank you. I'm very happy. I think I get the feeling that we're converging in a way.
Oh, good. Tell us how. Tell us how.
Well, I think some people are talking about the biology, the brain, the biomarkers, and so on. Another group is talking about behavioral and environmental conditions. Two things that we can observe and record in a lot of detail nowadays. So Julia was talking about the big data approaches. Marketing companies know so much about our behavior and our context, what we have around. And I'm from engineering, and engineers are developing new techniques to improve more of these observed type of data. And I think the fourth collection of information is the subjective experience. And I think that's where the clients, the users, the people who are suffering this are talking about. And I think there's an epistemological problem. We scientists are not generally comfortable with subjective experience because there is validity issues, there is reliability issues. So people cannot process user data as comfortably
They call it anecdotal. It's called anecdotal.
That's right. So scientists don't tend to use that anecdotal data in our studies because of those methodological issues. But I think that's a terrible waste. Now, if we can bring all these 4 bits together, we get a picture that is much more comprehensive. I think Paul put it very nicely there when you were talking about the client's experience. It's such a complex thing that you really need to understand what people are feeling. And the subjective experience is not just the client. I think the family's subjective experience is just as important. And we are approaching, or we are beginning to develop techniques that can help us understand that subjective experience.
But marketers do this every day of the week.
Yeah.
Why do we have to reinvent the wheel? This is not new.
Exactly.
They can tell you about people's psychoprofiles at the press of a button on their computer. Why are we struggling with this in biomedical research?
Well, there are 2 aspects, there. I think one, it's true that marketers have an idea of our psychology based on observed data. But that's not the same as subjective experience data. You can get some of that from focus groups and so on that marketers do. But the technology, the methodologies that marketing people use are quite sophisticated. Some of them look into subjective experience, but the majority really don't. So the big data projects that we're using are generally about observing what people do, what people buy, and so on. It's not about going and asking.
Psychiatric illness is a bit different from marketing for soap products or whatever.
Well, there is ethical issues, of course.
That's the problem. It can't be marketed per se.
It's not marketed, but there's methodologies for measuring people's behaviour and understanding their motivation.
Exactly. I wasn't talking about marketing.
I thought defending your position. I'm not very good at defence sometimes.
I thought you got the murderer off.
It's something to do with the fact that I don't understand what we're talking about. But
I think
they don't either. So.
If I give 1 million people an incentive to sign up to the Woolworths card or something because they're going to get discounts, and they're basically willing to sign over to this multi-billion-dollar company access to all of that data because they feel they've got an incentive. It's a very different situation to having to sit with an individual with an ethics-approved consent form and go through put all the structures in place that you need to with medical research data. And it's quite a very different way of approaching it, but I think maybe we need to try to learn from some of the ways.
Yeah. I just wanted to close briefly with a project idea, possible project idea.
Go on, this is radical. Go on.
Sorry. So something where you bring, in a longitudinal study, a cohort of people, and you look at subjective experience, could be some people at risk, and their families, and you follow them up, and you can do the brain, but you bring these 4 bits, not only the brain and the biology, the behavior, environmental data, but also the subjective experience. And in engineering, we have been doing behavioral analytics, the type of things that marketing people do for a while, and we are really good at collecting lots of data from individuals.
But let me challenge you on that, because there are suggestions there. Fiona's made one. But to what end? So that one day, in a distant land, a distant time from now, we'll come up with an observation which will either make a difference or not. Because there's a 5th thing that you haven't put into your data set, which is the care that they receive, the medications that they receive, the psychotherapy that they have, or the social support, which could have epigenetic phenomena in the brain, could affect the immune system that Mikhail's talking about.
Well, the way it's being used in engineering, there's an area called quantify self. So people is using this in a way as a therapeutic approach by looking and reflecting on my behaviors, on the way I feel. So there's already a number of people that is collecting this data for self-reflection. So I think even if it's not just for research purposes, even the aspect of people analyzing the data they collect is already, or can already be useful. Reflection can tell us why our psyche is working a particular way.
Thank you. Travis, and then I'll come to you over there.
Yeah, I think we're having some really fruitful discussion here. And I know we've sort of gone away from the initial proposition a little bit, but I think this idea of data and this whole idea of looking at not only the subjective consumer carer perspective, but also the clinical, scientific, biological perspective as well is really important. And I just had a couple of ideas around data, and Nolan actually just stole a couple of my points then that I've been waiting with. So I think it does come down to the big questions like what is mental illness? Is it just a set of biological neurons firing in our brain, or is it a real subjective experience in our situational environment as well? And for me, with data, I think Norman had a couple of great ideas about the data of the care people receive as well. I know myself, being a sufferer of depression and anxiety, have found that some clinicians and psychiatrists are real jerks. You can't connect with them. Some of them are lovely, such as Ian's wife, and and the same with.
Not Ian. It's not Ian.
And the same with medication. I doubt there'd be one consumer here that had found a medication with no bad side effects. I'm suffering side effects for over 5 years from my medication that have sort of screwed me over. But I'd like to know about all the other hundreds of thousands of young Australians here who have tried medications as well. What have been their experiences? Where's the data around that? You know, if I type in clomazepine or something on Google, you know, I might find a web forum, but where's the real data about consumer experience?
Thanks, Travis. So just to capture a theme that's coming through is massive integration of information, which is beginning to become possible with our computational techniques, including what you're talking about, you know, post-marketing surveillance of medications and getting true data, you know, accurate data about what happens to people out in the marketplace. Now, two of you there in the back have been very patient. I'll go to the person furthest back first.
Yeah. Is it working?
This is the other twin.
And then I want to come to Julio again.
Look, myself at the moment, I've got a cataract on my eye, which I can't afford to have done because it's not covered by Medicare. I'm living in a rather stressful situation, not through any fault of my own. I have just heard the latest budget, and I'd like to have our brain scans, everyone in this room, to see how our neurons and our little brains are going about dissecting what it actually means for us as individuals, because we're all affected by this budget in different ways. So it'd be very interesting if you had a brain scanner in here right now to actually test our brains, because I'll tell you what, it's a very cruel budget, and I think mental health is going to increase, mental illness rather, especially with youth. So how much can you scientists there, your neurobiologists, talk to the government about policies they make that are obviously,
so we're
talking about a subjective experience.
Can I just interrupt a second?
Yeah.
Tomorrow we are going to be talking about this, so it's good that you put it on the agenda, but tomorrow morning and afternoon we're going to be dealing with just these issues. Today, right now, I have to be disciplined and focused on the basic research and where that gets us. But what is very relevant in what you're saying is, can we afford what we come up with? If we do get the answers, can we actually afford to implement it?
Can we also afford not to have these people that are obviously in a field of experts? Like a village, we need this kind of village mentality, you know, where a whole village speaks and you're all together talking and sitting around the table and having the whole dinner together, not just a dessert at the end or an entree at the beginning and then somebody's invited to the main meal. I think it needs to be, we need to look at this whole thing.
Thank you. We're too close to lunch for that metaphor, but thank you for it. Yes.
So I would like to speak about this big data kind of information, and my expertise are on sleep. And I think it's definitely a major factor in many mental disorders. And one of the things that we are lacking is information, indeed this kind of information that we can get from the population. But I think technology has improved so much that we now have in our hands tools that are accessible to everyone, like cell phones, which can allow us to gather an enormous information, especially this kind of subjective information, that at some point, when it's big enough data, we can start making sense of it. And I think we should take advantage of these new technologies that are emerging and try to put them and integrate them in our research as part of the research programs.
So Jane Barnes is here. Where's Jane? Must have slipped out or doing an interview, because we'll get somebody to comment specifically on that who actually is working in this area. Julio.
Yes, actually, just in direct response to what you're saying, we are beginning a large new project to follow people prospectively using social media and cell phones and getting data like on a much more dynamic way than what we currently do. So I think what you said is very important. And the comment I had planned to do, I'm going to touch on sleep as well, which is that my comment is that, and it's a suggestion for a project as well. Of course, you know, there is something biological happening, and there is something, you know, happening in the brain, and we have to understand what that is and try to elucidate that. But there is, but I think that that's very confounded because most patients, I think nearly all people with mental illness, they fall into a vicious cycle that's like, it's not exactly the downward drift that was talked about in, I think, a very unfortunate setting. That should not be, people should not, just state that and offer nothing, but yeah, absolutely. But there is in reality a negative vicious cycle that the patients fall into. And once you are into that, the initial problem is so much more aggravated that whatever intervention you do is going to be very hard to bring the person back. So typically what happens is that the person, when they begin to have psychiatric problems, they're not going to eat as well as they should. So the nutrition goes down. So with the poor nutrition typically leading to obesity, then you have the fat tissue produce a lot of cytokines. So you have a situation of hyperimmunity, which the problem, if, as Michal and myself and many others believe, have an immune basis to begin with, obesity is not going to make it any better. If anything, it's going to make it worse. Then you have poor nutrition, poor sleep, disruption of circadian rhythms. Then that same person is not going to go on an exercise regimen like every day and go to the gym. So they are sedentary, and that only makes the whole metabolic cascade worse. Then, in addition, they don't, because of the nature of the problem initially, they don't focus so much on, productive activities. So they get more disengaged, both in terms of output and emotionally from others. And the more disengaged that they are, the more that they become. So it becomes really a complete downward spiral cascade. And I've been thinking about this a lot. Yes, we should come with a brand new biological treatment, but before we come up with that, or before we are ready to do that, why don't we try in a very controlled way what I'm suggesting as a boot camp for mental illness? So have people sleep, put them in a setting that they sleep 8 hours a night in a completely dark environment with no light clues, which has been shown to by itself cause depression. Then when they wake up, they have a nutritionally balanced breakfast. They have programmed activities. If they need medication, they have that in an optimally designed way for them. And then just structure the whole day like that. And I wonder if you get a person who is not doing well and you put them in a month of that very structured, regimented, and healthy setup, I'm sure that they will do better at the end, but it's something so simple and could help so many people, and we have not tested it. And it makes intrinsic sense, but nobody does it.
So it has been tested in anorexia and eating disorders.
Yeah.
So the Karolinska has a unit which does exactly that for eating disorders. So the question is, could you do it for
schizophrenia, for depression, for bipolar, yes.
So, sorry, Helen, you've been tackling the big data problem. Just very briefly, just give us some information on that.
Yes, thank you. This idea of using social media and using what we can get from the Twitter sphere and from Facebook and from other sources like LiveJournal and other blogs which are used by people who have a lived experience. What we're trying to do is bring that together and use that information to tell us a little bit about what's happening in terms of things like suicide, how suicide tweets move through the population, what is the pulse of the nation in terms of its mental health. And actually last night we have a program that we're running with Amazon and CSIRO, and we were actually and I can show you the data too, which is showing how emotion changed as the budget spoke. And this was every 10% of every tweet that was made. And what was interesting, actually, was that at 7:30 we had these huge spikes in anger, which just reflects back on. But of course the problem with this, and we are getting ethics approval to be able to do this, to link people's Facebook and their tweets and so on with their mental health data, which is largely through self-report. Ethics committees are allowing us to do that. This program of work is really quite accelerating quite fast. But it doesn't actually integrate with the sort of biomarker research and so on that's being done. So that clearly we have the technologies to do this. What we have to do is integrate it with the other things that Raphael was talking about.
Okay. I'm going to stop the group discussion here at this point. And our wonderful methodology that we planned for this discussion is already drifting a little bit off into the sunset. It is. So what I was going to do and panelists, don't go anywhere because I've got a job for you too. So the only people in the room that I really want to examine these projects and think about other things that we might do in a much more systematic way is the panel, because some of those projects are really technical. I haven't done you any service at all because I haven't gone into each one of those boxes there on that list and really explained what it is. You'll get a sense of it, and some of them come close to what we've been talking about, and feel free to look at that. But I think the discussion that's useful for you to have is what in general principles, what would be, if you like, the design principles of a big project that you've heard that really can make a difference? Don't worry so much about high risk, low risk. Just come up with, so I've heard stuff about, one of the things that we've heard about is massive integration of data so that we actually can link the fantastic work that basic biologists are doing with the world outside.
Norman, could you just define what you mean by make a difference?
I mean a track to treatment that will make a difference. That's what people care about, or effective interventions that will stop a disease process occurring.
I mean, all the discussion I've heard over the last hour or so, big data integration, how does any of that lead to better therapy? From my perspective, none of it does. Maybe for some disorders where, cognitive therapy and things like that have a big impact. Certainly some of the psychoses, some of the serious psychoses, cognitive therapy doesn't really help that much. And the comment we had about side effects of drugs, they largely come from the types of trials which are done at random, right? So, I'm just thinking of how do you drive therapy from any of this sort of data?
I agree that is the challenge. But you've also heard integration of what Cindy was talking about in discovery there through to rapid induction of trials. That is not necessarily a big data issue, but you may want more information to hand to actually understand why you're getting differences.
I guess, Cindy's point was pretty interesting because, it all came from brain samples, basically, right? So it all starts with the cell biology.
Clinical observations. I think it was really spurred on by this idea that there's sex differences and trying to understand the molecular basis of that.
But your target, right? How you pick to choose something.
Yeah, I didn't have time to go into it, but I think, one of the things that's being proposed is a national clinical trial structure. So that, and you can hang a lot of things off that. So you could do early intervention, longitudinally add the biologicals and have a clinical trial. So it's where you gain knowledge in an iterative approach. And so, in the reverse translation approach, like you say, you're going to have an arm that's going to do fish oil, Pat McGorry's favorite compound. But what are we doing in animal models? Like, do you know if treating fish oil is going to reduce anxiety phenotypes or molecular measures of anxiety in your rodent studies?
I guess my point is what Max made last night is how do you pick that fish oil, for instance, and not,
Well, we just agree. We just agree on, let's get started with 3 or 4 things that are, shown some promise and work on the molecular basis, like the SERM, the anti-inflammatories, the fish oil, and we just, gather the knowledge. And, it's not that important that we actually get the absolute molecule right first off the bat. I think what we need to do is set up the infrastructure and be working together and to set up the, the teams and get us, integrated. I think that would be a really positive outcome to say, look, we've successfully accomplished this. We've done translation, reverse translation.
Can I have a few words?
You can, Max.
Thank you. I think there are 2 kinds of ways forward that we've canvassed today and last night. One is extremely important in relating to what is referred to rather, I think inappropriately, as data collection. But to some extent, it was initiated, in my mind, by Pat McGorry and followed up a lot with the Brain and Mind Research Institute led by Ian Hickey. And that is, can we see in their case, in the adolescent period, subcategories expressed by different kinds of symptoms than we've acknowledged before of these grandiose terms like schizophrenia and bipolar. Can we get down to a much more fine-grained description of that immensely complex thing called human behaviour, let alone human behaviour that's suffering from one of these maladies. Now, there's a very large number of ways we can go about this at present, even in those that, shall we say, have the mature state of the disease rather than going through the adolescent prodromal ones. We need to know about the carers' opinions. We need to know, of course, about the opinions of those suffering. We need to know about the kinds of pseudo-objective measures of their behaviour with ambulatory devices, with smartphones and iPhones. We need all of that in order to try and capture something which the Diagnostic Statistical Manual has found beyond it in the last issue, and that is getting down to a much more fine-grained level. Now, the next point will be made, of course, is, well, so what? The so what is this, that there is an armamentarium of different kinds of pharmacological interventions which we are allowed to use in the context of humans rather than other animals, such as the fish oil, for example, or even vitamin D. And what emerged at the International Schizophrenia Conference last month in Italy was that some people have gone about looking at schizophrenia to some extent in that way, and they've found that some of the subcategories that we didn't realise existed are made much better with conventional approaches or with the use of pharmaceutical interventions which we're allowed to use under the regulatory regime. Now, if we just threw it at someone with schizophrenia, well, we haven't solved their positive syndromes, their negative syndromes, so it doesn't work. What this approach says, look though more closely at the behaviour. Maybe in the end, what we might come up with is a cocktail. One element, visual, for example, might hit this particular sub-syndrome component, another might hit this one, another one this one. And then we start to move forward collectively over the whole range of these subsystems. So I think the collection of the data in all sorts of different ways, Ian leads this in Sydney, Pat does it in Melbourne, all these different waves that have been canvassed today are extremely important in terms of trying to get a grip on the real human behaviour and what's gone awry in it. That's the first point. The second point is something which will not get a large clap from you at all, and that is this. This is not cancer. This thing up here consists of about 100,000 million neurons, each of which is connected to around about 10,000 other neurons, each one. If you add all those numbers up, you end up with something about 1 million times more complex than the human genome, which took us quite a long while to knock off. And even then, we've only knocked off about 5% of it because all the regulatory systems within the genome we haven't got a grip on at all. So don't kid yourselves that we're going to knock it off easily. Now, if you want to factor in endocrinology and immunology and other aspects of, shall I say, early physiology, if you want to knock that in, go for it. But you're not going to make the system simpler, you're going to make it more complex, of course, which might be the right way to look at it. That being the case, if you don't want to go down the stamp collecting, let's throw everything we can at the subcategories of a symptom, of a syndrome, like schizophrenia, which I think you should do, if you don't want to do that and you want to build up in a logical fashion the painstaking work of working out the connectivity of the brain in animal models and then comparing with all these fancy techniques, magnetic resonance imaging, spectroscopy of the brain, the axon measurements in the brain, etc., of human suffering in one of these subcategories, and then show that this animal that you've got has the same loss of white matter, the same loss of axons, same loss of neurons, same loss of various transmitter substances, gamma, and they match each other. Now you can take the animal and you've got some hope of throwing the entire pharmaceutical pharmacopoeia at it, because we're not under this huge regulatory regime where we're not allowed to throw these objects at human beings because we might kill them. I stressed last night in my dinner talk that virtually the whole armamentarium of pharmaceutical interventions, including clozapine, which is so extraordinarily remarkable in some ways in removing hallucinations, the positive effects in schizophrenia, all these were discovered in a 10-year period between about 1948 and about 1960. So what's happened since, as someone stressed earlier this morning, with the huge advantages in biology? Well, I think the only explanation I can get up for it is those guys, some of whom I corresponded with who made these wonderful discoveries, had no regulations on them. They can inject themselves, inject their patients, try this out, try that out, and some of their patients got more ill, and so they stopped doing it. Some of them got better. Very seldom, but they did say, oh, we're onto something here. We can't do that anymore. We've got to go back and use the animals, and then, in reverse translation, go back to humans. So there's two ways forward that I can see in relationship to this problem. One is the one of the subcategories of behaviour, both those who are suffering, the carers, the whole game, in order to try and get a much more close identification of what is the basis of all this, and then we might be able to use existing remedies. The other one is the slow but sure trawl through the workings of the brain.
Neil, and then Keith.
You want me to speak to you? Look, I just wanted to, I might take a little bit longer than it should, but look, I just wanted to perhaps finish, or not finish, but make a positive contribution in a different way. One of the biggest problems with neuroscience is that the one comment you'll always get as a consumer will be, oh, it's not quite as simple as that. So you go to them and say, well, you know, why is it so? And they say, oh, well, it's not easy to, you know, it's difficult and there's this and that, and the brain is very complicated. And I did read a book, because I'm involved in writing plays, and I've written many of them, but I read a book about the prefrontal cortex. And I was so proud of myself because I read this book and I understood at least 20% of it. And I spoke to the neuroscientists that I work with, they're all professors, and each one, 3 of them gave me a different view on the role of the prefrontal cortex in determining creative capacity. Now, what it said in the book was that a person with bipolar will have a more active prefrontal cortex, which gives them the capacity to absorb information because there's no inhibitor of information coming in, which is why you can get manic or depressed. That's very simplistic, it's more complex than that. So I thought, this is good, this is good, this is just what I wanted to hear, because this will help me with my writing and explains why I am the way I am. It's not just writing, it's my whole thought processes and whatever I am. So I spoke to all these and they said, no, it's not really the prefrontal cortex, it could be this, it could be that. So I thought to myself, well, I'm going to test this. And what I did was sat down with a friend of mine.
You didn't give yourself a lobotomy, did you?
No, I didn't do that. But what I did do was I got together all the information I could get on one topic on Indigenous soldiers in the Second World War, not just to Australia, all over. And together with my friend, we devised a play to write. So I sat down and I did it according to what the book said about collection of information and data, and I wrote the play in 2 weeks. It was not a bad effort. Full 1.5 hour play that we put on at Chapel off Chapel. The reason I tell the story is not to big note myself, although I would say it's not a bad effort, is to show that when you have access to the information that scientists are delivering up, even though they will say it's not as simple as that, it can give you great hope for what you're doing. And I think that I used to work with Paul, and we worked on the issue of, which is treatment-resistant depression. It is about giving people hope, and it ought to be. Otherwise, you know, it's got no meaning really. And in the same way as with the prefrontal cortex book, maybe it was dismissed as being not correct, but it gave me great hope of understanding of my own illness and, dare I say it, the capacity or the special capacities of people with mental illness. And I think that's very, very important. So that was what I just wanted to say.
Thank you, Keith. So, Keith, I want to Michal, and then I want to go to Norman Sartorius, and then I want to talk about what we're going to do next.
Okay, just briefly.
It gives me time to think about what we're going to do next.
Picking up on what Neil has just said, for the person with lived experience or their family member, the usual encounter is with a clinician. It's not with a neuroscientist. It's not with a researcher, it's with a clinician. And, you know, friends of mine have had this encounter and they've proposed to the clinician another way of treatment. And maybe it's not terribly approved officially, but it sounds like something worth trying. And do you know what the reaction is? Oh, no. We can't do that. We can't do that. It's all bad. The treatment we're giving you isn't helping, but we can't do that. That's where we are as patients and as carers. The response to any initiative from our side is, you can't do that. That's the problem.
Can I respond
Why
to
can't
that?
you do that?
I mean, I think that's a really important issue because coming from the perspective of a researcher that thinks they may have a novel treatment, trying to get psychiatrists to, you know, support their patient to go into the clinical trial has been very difficult. And actually, if I wasn't on Australian Story and was able to reach the televisions and the living rooms of the families, I wouldn't have been able to run my clinical trial because 90% of my patients came from the consumers and the families themselves. The psychiatrists were very resistant.
Michal.
One more thing I'd like to know in here is what about comorbidity?
Comorbidity, drug and alcohol.
Alcohol, self-medication. So how does that affect all this? Because that's a huge issue.
So let's take that on notice because it's a really important point. Michal?
I would like to bring back to the issue that we started 2 hours ago when you asked why we have so many drugs that were tested in animals and failed in the clinic. And you said, is it because the animal model is not correct, that we don't use the same model? I think that we have to go back to basic science, and the way to speed up the basic science is do a multidisciplinary type of work. And I think that we are now in mental diseases in a very similar stage that we were in in cancer research 20 years ago, when the first research on tumor were done in using tumor from human and implanted it into animal. And we used at that time immune-compromised animals, so the tumor will not be rejected. And a drug was found, and when it tested in a human, it didn't work. So I think that we have to go back to animal model, and we don't have to reach the same animal model, but do several animal models. So one animal model, of course, will not be the ultimate model. Every tested direction or compound should be tested in several animal models.
In parallel.
Yeah, in parallel by multiple centers. I think that we need a basic understanding of the disease. It's a heterogeneous disease like all other diseases. It's not a unique situation.
So Michal, to do that requires quite significant change.
I agree.
Because people are so committed to their model.
I agree.
Now, what's the sea change that needs to occur behind all this? Is it the war on mental illness that we're going to create such an impetus that we're going to demand that labs in Rehovot and labs in New York and labs in Sydney and Melbourne do cooperate with theirs, and they take new molecules and test them in parallel, and they commit to that? What's got to happen?
I think
Private sector doesn't even talk to the public sector as well. They don't know how to do it.
it needs a lot of thinking before I'm giving you an answer, but I think that quick and dirty clinical trial will not work. We need basic understanding, and that's where we are, most of us. Those of us, we need really thinking out of the box, as many are thinking, as I proposed before. It's not a disease of the brain only. And I think that we have to remember that most of the drugs in animal model were tested before the onset of the disease in many cases. And we have to appreciate that early diagnostic is very important. treating before onset and treating after onset is totally different. So I think that we need a paradigm shift in the way that we approach the research rather than doing a quick and dirty.
But would you put all your money on the animal models, the absolute commitment to preclinical testing, when you might get to the point, I mean, Cindy's point would be, maybe you do want quick and dirty just to see whether or not you've got proof of concept in humans that might go on to a bigger trial, and it's not one or the other. You might do
It's not
both.
one or the other. I think that we cannot skip the preclinical studies, the basic biological research.
But the only advances to date in psychiatry have been, you know, quick and dirty clinical trials, you know, the proof's in the pudding sort of thing. And there is some evidence in brains in animal models, like in neurology with Alzheimer's or Parkinson's, where you get traction in animal models and then they fail in the human. I mean, the rat isn't a mini human, and that's been a problem.
I agree with you, but you can, from the basic science, you can go back and forth to the post-mortem brain and many other parameters that you can compose. So you don't have to jump just from the animal to the patient. There are interim phases that you can test in post-mortem samples or in blood samples.
Right, but I don't think it's one or the other. I think we need to advance these in parallel.
Yeah, Cindy, I mean, I think, you know, I mean, our understanding of neural function is really right at the scratching of the surface. I mean, to go from animal to humans right now is difficult, but I think you have to go down that track because ultimately that's where the disorders are.
Yeah, but you don't want to wait 50 years for do a new drug.
We've waited 60 years so far with nothing. So
Can I just try the
that's
tyranny of
because
the
the drug
microphone?
companies have been doing Me Too variations on D2 receptor blockers.
Yes?
Sorry, just a small contribution here. A lot of the discussion about animal models translating or not, there are points. You look at a mouse brain, certainly doesn't have prefrontal cortex. It's quite different from the human. If the problem isn't prefrontal cortex, you don't see it. But more recently, there's been substantial progress in developing new animal models, knocking knockout genes in animals that do have prefrontal cortex. This is just like last November appeared in the journal Nature. So there's a lot there of potential for, if you target to the right animal models, you might actually understand what's this switch. You can talk all of you want about risk factors and genetic markers, but the fact is the person has a genetic marker that may or may not undergo this switch. So if you really want to find what's the Helicobacter equivalent, you would have to go and look at those animal models.
can I comment on something which I think is rather delightful in this context, and that is that the guys coming back from the war zones, about 20% of them have post-traumatic stress disorder. The major way of treating them is using extinction tests. The extinction test was discovered in 1934 by Pavlov using dogs.
Sorry, go on.
With talk about efficacy and where would you put all your dollars, I want to bring it back to the human individual experience that is mental illness, and that it would be great to put the dollars where it's going to alleviate the most suffering in the quickest possible time, and actually to ask consumers and perhaps their family members who are also affected. The lived experience, I think, is missing today, and it feels really weird coming here as a rank outsider. I couldn't understand about 20% of the paper you gave me, so it would have been great to have a few footnotes just explaining some things. But I'm feeling like the person is missing, and I guess the advantage of having a person over an animal model is that you can actually ask them. You can develop a relationship with them, which is what's missing a lot in psychiatry, and I would love neuroscience to have mapped my recovery journey across 20 years from childhood trauma and to see the efficacy of that.
So I think that point has been very well made, and that challenge to integrate that information is one that's really come up front and centre to the information that's been developed in the laboratory. There are a couple of issues which we've only just alluded to, which might need a little bit of elucidation. One of the problems with jumping to the quick and dirty is new regulation. It was much easier to do drug trials in the bad old days when we actually didn't have good ethics committees, which allowed prisoners, American prisoners, to be experimented on without their permission.
So I'm challenging that and saying that it may not need drugs. It may need interpersonal relationships.
Sure, it might need other things, but to test new things more and more, ethics committees require you to have made sure it's safe in animals, and there's a whole series of things, which is why Max argues it may well be one of the reasons why you actually haven't had new drugs in mental illness, except that you say it's not all drugs, but we haven't had very many new ones, if any. And one of the reasons is the different regulatory structure and ethical structure for doing this.
So I really like Julio's comment about having a boot camp for sleeping, and I wonder how many people here without a diagnosis would survive well without sleep. The situation he describes sounds like the stable early childhood that me and my colleagues missed out on, and I'd love it. Thank you.
Thanks. Norman.
Well, I was admiring many of the presentations and the interest with work, and I'm grateful for the moving testimony as well as for the information from the people who experience illness. But I wanted to ask you, do we accept Proposition 1 or not?
Very good question. I didn't hear much difference in the panel from it.
Oh, yes. I don't.
You don't agree with it?
No, I don't think that you should just track the adolescent. I think you should actually move in with a fully formed, card-carrying illness itself. Because at the extremes of the spectrum, you might be able to pick up more easily the sub-syndromal aspects of it at the very minute levels of behaviour and start to see whether, in fact, some elements of that can be helped by a whole lot of compounds which are not under the regulatory prescription at present. So I think we ought to look at the fully fledged card-carrying disease.
So which is why I'm going to change the agenda. First of all, I don't think we gave you enough information. I think you made a reasonable point there, but we didn't give you enough information about the list of things that are on that list. I don't think we're ready yet. There's too many ideas. It's not too many. There's a fantastic number of ideas floating in the room. And in fact, they are coming together a bit. I think Raphael's right. There's a bit of convergence. I think it would be, so if I threw you to small groups now to talk about what are their projects, I think it's just too early. I was thinking, what would I think about if I was asked to do this? So I apologise to our volunteers because they're all ready to go and start. So what I propose is that the natural flow from this discussion is into Proposition 3. Because Proposition 3 leads into what Max is talking about, which is people with established illness moving forward. So we have a partial acceptance of, and one of the reasons why we not universally accept Proposition 1 is that Proposition 3 is relevant too. And what I think we should do is go into that discussion. So people who are on the panel for Proposition 3, gird your loins, because you're coming on next, not the physical health one. We'll do that tomorrow. Because I think then we'll start to crystallize some ideas quite well, and then we can have a much more useful discussion. And it may be much more generic than the specificity of what's on that list. Because I think what Ian and others can do afterwards is match what we've come up with that list, or come up with stuff that's new. So I think we have to describe the elements of what we'd like to see about a leap forward, and then work on that. And I think we'll be more ready for that discussion after we've had Proposition 3 discussed. Are we cool on that?
Absolutely.
Could you please thank our brave panellists?